Good afternoon, everyone. Welcome in today's class. Let's begin with the one more topic of biochemistry, that is heme synthesis and the porphyrias which are related with each and every step. So we'll be doing this. So let's begin with today's class, and as we say in the beginning, let's crack NEET PG together. So the topic of today is heme synthesis and the porphyrias which are related to heme synthesis, right? So, before beginning with the topic, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platform for the preparation of NEET PG examination, and at Unacademy you will have the best educators all over India. So you can get the benefit of being mentored by the best faculty member all over India. Then there are special classes with Unacademy, and you can ask your questions and queries and doubts directly with your faculty member, and that is why they are called as special classes, because the faculty members, they'll be solving their queries, solving your queries, right? Then there is this test series, and in this test series you can go through the free mock test, weekly test, and for any kind of examination which you want to qualify, it is very much important that you first of all know the pattern of the examination, and you are very much used to with the questions which are going to be asked. So test series are really very helpful in the preparation of examination. So that is also possible with the help of Unacademy. Then you can just go to Play Store and write Unacademy, and Unacademy Learning app will be shown, and you can just download the Unacademy Learning app and make your account and log in, and very good to go for the preparation. Then it is very much flexible that you can prepare for the examination anywhere, anytime, any topic which you want to prepare. You can just log into app and you will have the immense knowledge of all the video lectures. Then language can be of your choice, because there are many faculty members who even teach in the regional languages, so that can be one of the added benefit. Then unlimited practice is the case with Unacademy, because if once the quiz is taken or quiz was conducted, it can be retaken as many number of times as one wants to. And the video lectures, they are recorded, so if they are recorded, it means they are available for a long duration, and you can anytime go and check out the video if once you have learned from that video and you are feeling that yes, the topic is quite interesting by this faculty member, so you can anytime go back and go through the lecture once again. Then you won't feel anything different from a normal classroom, because you can chat with your educator. Then there will be interactive polls, and in these interactive polls there will be n number of questions, and these questions and in these questions you will have to attempt one of the option, and you will also come to know that who all are feeling that yes, these options are also correct if actually they are not. So you will also know the mindset of students, that where they actually get confused, and faculty member, they'll also be telling you about each and every option, and you will understand in very precise manner that which option is right and how to attempt the question. So it will be very much fun, as well as it will be very knowledgeable. Then learn anytime, anywhere. If you are not in town, you are holidaying or anything, then also you can just attend your classes anywhere. So learn anytime, anywhere does not make it a very crude type of thing. You can just learn anywhere, right? Then there's a plus platform. So on this plus platform, you will get all the 19 subjects, each and every video in the recorded manner, and there will be short-term courses and crash courses which are being conducted timely for the upcoming exams, and there will be motivational videos as well as the strategies to qualify the examination. So the plus subscription can be very much helpful in qualifying the NEET PG examination, and you need to pay a certain amount for this subscription. So the amount is very pocket friendly, and if you're going for a 12 monthly or a 24 monthly subscription, then it will be very much helpful to you, because then you can make the most out of all of the videos which are available on the platform. Otherwise, in a very short duration, there is immense and immense of videos and data available, you won't be able to do in a little less time, but if you are going for a 12-month or a 24 monthly subscription, you can easily cover each and every topic in a detail and in precise manner. Then in this plus subscription, you can get an additional 10% discount if you're using my referral code, that is FB01. So don't forget to use my referral code if you're going for plus subscription. And let's begin with the class. The topic of today, that is heme synthesis. Heme synthesis. So there are n number of intermediate in this whole process, and it's a quite lengthy one. There will be lots of structure and there will be lots of processes. So what I've done for this heme is I've made a mnemonic so that you can very easily understand that how the heme synthesis occurs and what are the intermediates, because many a times we remember the name of the intermediate but we don't know what is the sequence. So I have made a mnemonic and we can know the sequence also. Hello Sam, welcome to today's class. Now what is the site of heme synthesis? The site of heme synthesis is erythroid precursor cells. This is the site. Then this is the site where major synthesis occurs. The rest of the remaining synthesis occurs in liver. So erythroid precursor cell and liver. If these are the major cells, the organelles which are involved in the synthesis is mitochondria and cytosol. The two organelles which are involved in the synthesis of heme is mitochondria and cytosol. Right? Some of these steps will occur in mitochondria and some of these steps will occur in cytosol. So let us now talk about the mnemonic and then we'll be moving towards the whole process. So I'm telling you the mnemonic over here because whenever we're doing some step, I'll be telling you that the initial of the mnemonic, how is it related to the actual intermediates, right? So let us just read that. Some great designers participate hard under carpenter's procedure, perfect home. So we'll be taking the initials of the each of the word I have written over here, right? So let us begin with the actual process of heme synthesis. So first of all, what happens is CoA, succinyl CoA reacts with glycine, or it's a kind of condensation reaction. It's a kind of condensation reaction. Condenses and it produces a product that is called as delta amino rivulinic acid. This is the increase the voice a little bit. Okay, I'll just talk a little loudly. So the product that is forming by the condensation of alpha from the condensation of succinyl CoA and glycine, we'll be getting delta amino levulinic acid, delta ALA. Now the enzyme which is actually doing this process, let us write the name of the enzyme. The enzyme is ALA synthase. ALA synthase. This is the enzyme which is performing this kind of condensation reaction. And if it is a condensation reaction, what will happen? Some of the things will go out. So what are the things which are removed from this? The things that are removed is CoA and carbon dioxide and sulfur hydryl group. So these three things will go out in this process, and this enzyme is PLP dependent, that is pyridoxal phosphate dependent enzyme, and this pyridoxal phosphate is actually required for activation of glycine. Activation of glycine. I hope now I am audible to everyone. Okay. So delta amino levulinic acid is formed, that is delta ALA. And how are we going to relate it with our mnemonic? We said sum for succinyl CoA, great is for glycine, and designers is for delta, right? So we will just write small mnemonics also over here so that you can remember the things now. So S is for sum and G is for great and delta is for designer, right? So we are good till now. Let us move ahead in the process. After delta amino levulinic acid, there will be removal of one water molecule, and the process is a kind of dehydration reaction. That was first one was condensation. This is dehydration. So the enzyme which is participating in this process is ALA dehydratase. Dehydratase. This is the enzyme which is performing this reaction. And what is so special about this enzyme is it is a zinc containing enzyme and it is inhibited by lead. If there is lead, then it will be inhibited. So in any of the case, if there is a presence of lead or there is the deficiency of zinc, in both of the cases, it will lead into anemia. In both of the cases, it will lead into anemia, right? And if we are saying that delta amino levulinic acid is participating in some kind of dehydration reaction, so how many molecules of this particular thing? So two molecules of delta amino levulinic acid will condense, or we can say it will dehydrate, and it will lead into formation of a product which is called as porphobilinogen, or in short form we can write PBG, right? PBG. Now this P over here for our mnemonic, this P is for participate. P is for participate, right? Let's move further, that this is formed. What is its structure actually? If it is formed, what is its structure? Because from here, the things will be quite dependent on the structure also. So the structure over here is monopyrrole ring. This is the structure of monopyrrole ring, right? So porphobilinogen is formed by dehydration of two molecules of delta amino levulinic acid. Now these four molecules of porphobilinogen, they will react in a process which is a kind of condensation reaction. They will condense together and there will be removal of four amino groups. Now the enzyme which is doing this kind of reaction, let's learn the name, that is Porphyrianogen synthase. This is the enzyme which is actually participating in this reaction. There are synonyms of this enzyme, that is PBG or porphobilinogen deaminase, right? If you can see, there is removal of four amine groups, so it is deaminase, and the another name is HMB synthase, the product which will be forming. So we haven't yet written the name of the product which will be formed. So the product which is formed from this kind of condensation is HMB, that is hydroxy methyl bilane. This is the product formed after condensation of four molecules of porphobilinogen, right? Now if we are saying that this is the product which is formed and this is a single pyrrole ring, and if four pyrrole rings they are condensing, it means the four pyrrole rings, they will be joining together. So how they will be joined? They will be joined in this ways. They will be joining in this ways. So this is called as linear tetrapyrrole. This is the linear tetrapyrrole. Now, whenever we are reading, we are also talking about the enzyme. The very first enzyme that we saw, ALA synthase, what did we say over there? That it is PLP dependent or pyridoxal phosphate dependent. Then second enzyme we did was ALA dehydratase, and we read that it is a zinc containing enzyme and it is inhibited in presence of lead. Now let us talk about this third enzyme. What is so different about this enzyme is that in its deficiency, if this enzyme is deficient, or in its deficiency, what will happen? Acute intermittent porphyria will occur. Acute intermittent porphyria. Acute intermittent porphyria. Right? This will be caused, and it will be manifested by abdominal pain, and it will be manifested by neuropsychiatric manifestations. So these will be the symptoms. So in the deficiency of uroporphyrinogen synthase, there will be no formation, or there will be little formation of hydroxymethylbilane, and it will be manifested by acute intermittent porphyria, right? Let's move ahead from here, from HMB, that is hydroxymethylbilane. And hydroxymethylbilane, the mnemonic which goes for hydroxymethylbilane is your hard. Participate hard, right? So hard. And now the next mnemonic that we read is under, right? So the next product that will be formed from HMB must start from U, because we are saying under, right? So let us write HMB and let's continue the process. That is HMB. This HMB, it is a linear tetrapyrrole. So now it will undergo cyclization. It will undergo cyclization and it will form another product that is called as uroporphyrinogen. Will form uroporphyrinogen, and the mnemonic that goes for uro is your uroporphyrinogen is under, right? This thing. Now coming back to the enzymes which will be actually involved. So the enzyme responsible for formation of uroporphyrinogen is uroporphyrinogen 3 synthase, co-synthase. This is the enzyme which is participating in this type of reaction. And when we are talking about the structure, that HMB is a linear tetrapyrrole, and when we are saying the cyclization will occur, so how the uroporphyrinogen will actually look like, and it is actually uroporphyrinogen 3. Why it is 3, that also I will tell you. This is the structure of uroporphyrinogen. So the groups which are present at these ends are AP, AP, AP and PA. A stands for acetyl group, and P stands for propionyl group. Propionyl group. These are the groups, right? So we are saying that here it is 3, uroporphyrinogen 3. Can you see the sequence? That firstly we are saying AP, then we are AP, but for this we are saying 3. If it is also in the sequence of AP, then it will be uroporphyrinogen 1. But here it is PA. It means that it is third in the series and it is the given numeral 3. Right, now we are also talking about the deficiency of the enzyme, or specificity about that enzyme. What is so great about that enzyme? So the deficiency of uroporphyrinogen 3 co-synthase, it causes the disease congenital erythropoietic porphyria. Congenital erythropoietic porphyria. Let's write the full form here. Congenital erythropoietic porphyria, right? So we have understood till here that what is happening. Now let's move further. Now what will happen further in this reaction? that there will be removal of 4 carbon dioxide molecules in the presence of enzyme uroporphyrinogen decarboxylase. And here it is uroporphyrinogen, the same thing. So uroporphyrinogen decarboxylase, 4 carbon dioxide will be removed, and the thing that will be formed will be coproporphyrinogen. This is the product. So after under the mnemonic that we have seen is carpenter. Work hard under carpenter. So U is for under in our mnemonic, and C is for carpenter. Right, so this is our mnemonic. Now, how the structure will look like if uroporphyrinogen is having this structure, then if there is removal of 4 carbon dioxide, how the structure will change or the groups will change. Here there was acetyl group, this was propyl group, acetyl, propyl, acetyl, propyl. Now the acetyl groups will change into methyl groups. So it will become MP, MP, MP and PM. So in place of acetyl group, methyl group is coming. So M is for methyl groups. Right. So we have understood how the structure changes and what is the mnemonic. Now for every step we are also discussing the porphyrias. So the porphyria which is associated with coproporphyrinogen, is the Porphyria cutanea tarda. Porphyria cutanea tarda. If there is deficiency of uroporphyrinogen decarboxylase. Now deficiency. We should also mention deficiency of uroporphyrinogen decarboxylase. It will lead into a porphyria that is called as Porphyria cutanea tarda. And what is so special? It is the most common porphyria. Now, if it is the most common porphyria, what kind of porphyria it is? It is a kind of cutaneous sensitivity. Towards what? Photosensitivity. That too, photosensitivity. Right? Cutaneous sensitivity, photosensitivity. And why does this photosensitivity is occurring? Because there will be release of lysosomal enzymes. There will be release of lysosomal enzymes and specifically hydrolases. Hydrolases will be released and it will be causing Porphyria cutanea tarda, and it is a kind of photosensitivity because of the release of lysosomal enzymes and specifically hydrolases. Right? So deficiency of uroporphyrinogen decarboxylase is the reason of Porphyria cutanea tarda. Right? So this was the porphyria. Now coming back to the chain of reaction. So don't worry if you are feeling that we are doing it in parts. We'll be just making a summary of everything. And it will be very interesting for you to learn in that ways also, because now you also know the our mnemonic. So it will be very easy. Now let us take the process from the formation of coproporphyria. Coproporphyrinogen. Right, and this is coproporphyrinogen 3. Now what will happen, that again there will be removal of 4 carbon dioxide molecules. If they will be removed, then what will happen? Let us see. There will be removal of 4 carbon dioxide molecules in presence of which enzyme? Coproporphyrinogen porphyrinogen oxidase. And this enzyme is present in mitochondria, the mitochondrial enzyme. So this step occurs in mitochondria. So what will happen? Now what thing will be formed? Protoporphyrinogen. Protoporphyrinogen 3 will be formed. So see over here, we have already understood that C over here is for carpenter. P over here will tell you about procedure, or in the mnemonic it is telling you that remember the sequence procedure, right? And if it is a specific kind of structure, so let us draw the structure also of protoporphyrinogen. The structure will be like this. If we are saying that coproporphyrinogen is MP, 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 then here it will be MV, MV, MP and PM. Two of the propyl group will get converted into vinyl. V is for vinyl. Only two places it will be vinyl group, and that will be because of the change of the vinyl group here. Propyl group will change into vinyl group, right? So I hope you people are understanding. Please write in the comment box if you are understanding the process well. Now, there will be another enzyme, that is protoporphyrinogen oxidase. Protoporphyrinogen oxidase. What it will do? It will change the protoporphyrinogen into protoporphyrin. When it will change it into protoporphyrin, this P over here, it tells you about perfect, procedure perfect, right? So this tells you about perfect. Now the last mnemonic that is left is home. So lastly, it will lead into formation of heme, right? And H over here tells you about home. Tells you about home. H is for home, right? So the enzyme which is responsible for this reaction is ferrochelatase, and there will be incorporation of but obvious ion. Incorporation of iron in its ferrous state. Incorporation of iron will be done over here, and it will take the central position of this whole thing. It will take the central position here. So this is how your heme will be formed. This is how your heme is formed, right? And heme is for your home, heme or home. So hope you have understood the process. We'll do the summarization of whole thing. This was the detail, and we'll now do the summarization. So I hope you have understood all the process along with the mnemonics. Let us move further to the summary. Okay, summary along with the summary. So how we will summarize this thing? Succinyl CoA will react with glycine, and it will form delta amino levulinic acid, and it will form porphyrobilinogen, and it will form hydroxymethylbelane, and it will lead into formation of uroporphyrinogen 3, and further it will lead into formation of coproporphyrinogen 3, and coproporphyrinogen 3 will lead into formation of protoporphyrinogen 3, and from protoporphyrinogen 3 it will lead into formation of protoporphyrin, and finally it will lead into formation of heme. So how this thing is happening? Some great designers participate hard under carpenters, procedure, perfect, home. Right. So we have understood the sequence well. You can just write this and you can elaborate the sequence. We'll also discuss about the enzymes which are present at each and every step. So now let us talk about the enzyme which is doing the conversion. So the enzyme over here that is responsible for this process is ALA, ALA synthase. Right. And this is a rate limiting step. It's a rate limiting step and it is inhibited by feedback inhibition. This ALA synthase, it is a rate limiting enzyme and it is regulated by feedback inhibition. Now, the conversion of second step, it occurs with the help of ALA dehydratase. Dehydratase, and this dehydratase, it is inhibited by lead. It is inhibited by lead and it itself is a zinc dependent enzyme. Now conversion of PBG to HMB, it occurs with the enzyme that is uroporphyrinogen, uroporphyrinogen synthase. Uroporphyrinogen synthase. So there is an porphyria related to this. We'll be doing this porphyria. Also, one more time, then HMB is converted into uroporphyrinogen with the help of enzyme that is uroporphyrinogen co-synthase. Co-synthase. And this U3 will be converting into coproporphyrinogen with the help of enzyme uroporphyrinogen decarboxylase, right? And this C3 will be converted into P3 with the help of coproporphyrinogen oxidase, and this P3 will be converting into P with the help of porphyrogen oxidase. And lastly, the enzyme is ferrochelatase. So this is the summary of whole process along with the initials so that you can very well remember the things. Now let us move to the porphyrias. Porphyrias. So first porphyria is because of the deficiency of ALA dehydratase. It is because of uroporphyrinogen synthase. Just a second. Uroporphyrinogen synthase. Uroporphyrinogen synthase, the first porphyria, and it is called as acute intermittent porphyria, and it is the acute disorder. Acute disorder, most common one, and it is manifested by abdominal pain and neuropsychotic manifestation, right? So the name of the porphyria over here is acute intermittent porphyria, and it is caused because of the deficiency of uroporphyrinogen synthase. Now moving towards the next porphyria, the enzyme which is actually responsible for this porphyria is uroporphyrinogen co-synthase, right? Uroporphyrinogen co-synthase. So the porphyria which is associated with this is congenital erythropoietic porphyria, CEP, congenital erythropoietic porphyria, and it is a kind of autosomal dominant porphyria. Autosomal dominant porphyria. right? And rest all of the porphyrias are autosomal recessive porphyrias. This one is autosomal dominant porphyria because of the deficiency of uroporphyrinogen co-synthase, right? Let us move to the next. That is because of the deficiency of uroporphyrinogen decarboxylase. Decarboxylase. Deficiency of this causes Porphyria cutanea tarda. Porphyria cutanea tarda, and it is the most common type of cutaneous manifestation or most common type of porphyria because of the photosensitivity, because of the deficiency of this particular enzyme, right? Then coproporphyrinogen oxidase, it is a kind of mitochondrial enzyme, and here only let us talk about the porphyria, that is coproporphyrinogen oxidase. It is a kind of mitochondrial enzyme, right? So it leads into hereditary coproporphyrinogen porphyria, right? So four porphyrias we have seen, that is acute intermittent porphyria, then we have seen congenital erythropoietic porphyria, then we have seen porphyria cutanea tarda, and then we have seen hereditary coproporphyria. Now there are two more porphyrias which are left because of the porphyrinogen oxidase, porphyrinogen oxidase, and its deficiency leads into variegate porphyria. It leads into variegate porphyria. Variegate porphyria, right? And lastly, because of ferrochelatase deficiency, the porphyria which occurs is protoporphyria. Protoporphyria. Okay, so we have seen total acute intermittent porphyria, congenital erythropoietic porphyria, porphyria cutanea tarda, then hereditary coproporphyria, variegate porphyria, and protoporphyria. So we have seen 6 porphyria in the heme synthesis, right? And we have learned about the whole heme synthesis quite in detail. We can also further see how the heme is degraded in the further videos. So that also we will do in the coming days, how the heme is actually degraded. This was how it is formed, and the porphyrias which are related. We will also see porphyrias more in detail in some other video. Here also we have seen almost all the porphyrias which are related, and along with the enzymes which are responsible. So all the things which are actually important for PYQ we have discussed over here. So this is it for your heme synthesis. Hope you people have understood the heme synthesis in detail, and hope you will like reading it further. That is its degradation and some porphyrias. And we'll meet in our next class tomorrow. We'll be doing some another interesting topic of biochemistry. So just keep yourself updated about the upcoming videos. And if you really like my video, please give me a thumbs up. It really motivates me. And please subscribe our channel, that is Let's Crack NEET PG. And also use my referral code, that is FB01, for an additional discount of 10% on the Plus subscription. So by getting a 10% discount in Plus subscription, it will be quite helpful. And please subscribe the channel and like my video, and come back for some other great interesting biochemistry videos which we'll be doing together, and we'll be making it simpler, easier, easy to learn with some mnemonics, right? Okay, thank you very much everyone. We'll be meeting again. Bye.